Dear Global Soil Biodiversity colleagues, welcome to this GSBI webinar on biological soil crusts. Good morning, good afternoon, good midnight, wherever you are. We have one announcement to make at the start. This session will be recorded, so if you have no problems with that, then keep going. And yeah, this year it was quite a heavy year for us. We have lost Diana Wall, as you know, and we really had to repack and see how to get things going, to keep things going. And since we are sure that Diana would have wished us to continue, this is what we are very much determined to do. And so this is also what is happening with the webinars. We continue with the webinars. They were really successful. Diana has set them up during COVID time. And so today we have the webinar on biological soil crusts. And I'm very happy to announce Raúl Ochoa from the University of Cádiz. Hello, Raúl. And,
hello, Bim.
Hello. And Raúl will chair this meeting on soil crusts. So Raúl, the floor is yours and take on control.
Thanks, Bim, for the introduction. Good morning, afternoon, and evening, everyone. Welcome to the seminar on biocrusts organized by the Global Soil Biodiversity Initiative. My name is Raúl Ochoa-Hueso, and I am a professor at the University of Cádiz in the south of Spain. Well, first of all, I would like to start by thanking the GSBI committee for inviting me. It is my pleasure and a real honor to act as a moderator today and to be able to introduce some of the most notable world-leading researchers on biocrusts. The seminar of today will consist of 4 presentations of 5 to 6 minutes, followed by Q&A. During their presentations, you can type in your questions in the Q&A box, and we will do our best to answer as many as we can at the end. But before we hear their talks for the next half an hour or so, let me briefly introduce you to the topic. Biological soil crusts, or as they are more commonly called nowadays, biocrusts, form miniature ecosystems that result from the intimate association of soil particles and photoautotrophic and heterotrophic organisms such as cyanobacteria, fungi, even algae, microfauna, lichens, and bryophytes. These organisms live within or immediately on top of the uppermost millimeters of soil, which is why many see them as the Earth's living skin. Since the early works of biocrust pioneers like Jane Belknap and Otto Lange, researchers have shown that the distribution of biocrusts is usually conditioned by the presence of a continuous layer of vascular plants, with which they cannot compete. This is the reason why they're typically found in water-limited ecosystems, such as hyper-arid, arid, and semi-arid regions, including the Negev, Namib, and Mojave deserts, the Colorado Plateau, and the Mediterranean basin. Biocrusts can also be found in alpine and polar regions, including the extreme environments of Antarctica, which are characterized by the frequent lack of liquid water and sparse vegetation. Soil is another factor that may condition the distribution of biocrusts, with fine texture soils often supporting a higher cover of lichens and mosses. We can also find biocrusts as early successional communities in more mesic environments, where they play key roles as ecosystems engineers. They also pioneered the colonization of the surface of our planet more than 500 million years ago, enabling the evolution and development of more complex communities, eventually leading to the wealth of terrestrial ecosystems that we observe today. Well, apart from their colorfulness that partly derives from their huge taxonomic diversity, comprising organisms belonging to a wide variety of domains, kingdoms, and phyla, why should we be talking about biocrusts today? Well, I think that we should do so because despite being small in size, where they are prevalent, biocrusts contribute disproportionately to ecosystem functioning and services. For example, they stabilize the soil surface, thereby preventing erosion. They enhance soil fertility by fixing atmospheric carbon and nitrogen, and they influence soil hydrology. Biocrust distribution is also affected by vegetation dynamics and the microenvironment that plants create. While they cannot compete with vascular plants for resources such as light, nutrients, and water, favorable microsites under shrub or tree canopies may enhance biocrust development by increasing soil moisture and fertility, indicating a very complex interplay between biocrusts and plants, which is reflected in their highly heterogeneous distribution across the landscape. Well, this makes it particularly challenging to model their distribution across spatial scales, from local to global. And finally, in addition to vegetation and climatic and edaphic properties of the habitat, physical disturbance, as well as the effects of ongoing global change, have a strong effect on biocrust growth and the types that may occur. This raises the question on how to restore these communities where they have been lost to recover their functions. Today, we have 4 excellent speakers that have greatly contributed to the development of the field. They will help us navigate the latest advancements in biocrust research. First, we will listen to Bettina Fifa. Hi, Bettina. You can say hello. Bettina.
Hi,
we'll talk
Raúl.
Hey, Bettina.
Everybody.
Morning.
Good morning. Good afternoon.
So, Bettina will talk about the role of biocrusts in the Earth system processes and the latest advancements in biocrust mapping. After Bettina, we will have the pleasure to listen to Emilio Rodriguez-Caballero. Hi, Emilio.
Hi, Raúl. Hi, everybody.
Well, Emilio will introduce us to biocrusts from a multiscale perspective, thus helping us to understand how biocrusts are organized from the local to the global scale. After this, we will zoom in again by the hand of Claudia Colesie. How, Claudia? Hi, Claudia.
Hello, hello.
Hey. Claudia will tell us about one of the most extremotolerant communities on the Earth, the biocrusts from Antarctica. And finally, the one and only Matthew Bowker. Hey, Matt. Yeah.
Hi. Thanks everyone for joining.
I think I'm not mistaken if I say that Matthew has inspired many old and young biocrusters, and that today will keep inspiring us by introducing us to the fascinating world of biocrust restoration. And without further ado, our first speaker, Bettina Fifa. Bettina is professor of botany at the University of Graz in Austria, and she seeks to understand the composition, distribution, and functional relevance of biocrust communities. She has published seminal articles on biocrusts that have been instrumental to redefine the field and to spur further research interest. Bettina, please, the floor is yours.
Thank you very much, Raúl, for this kind introduction. Yes, and thank you very much for the opportunity to speak here. So my talk will be on the role of biocrusts in Earth system processes. But before we dive into that, I want to give you a short glimpse about our research agenda in my group. So our research is on cryptogamic communities in general. These are, on the one hand, cryptogamic communities on soil, forming biological soil crusts, but we are also interested in cryptogamic communities growing epiphytically on trees and also colonizing rocks. So we are interested in who is there, and to find, wait a second, I look for a pointer. So, and if you are interested in the methods we apply, you have to look here on the right-hand side. So we are interested in who is there in these communities. We are interested in where do they occur, so both the geographical location and the habitat conditions where our communities occur. We want to know what they are doing and how they are doing that. These are biogeochemical cycling processes like water, carbon, and nitrogen cycling. Also, we are also interested in the emission of volatile organic compounds and the emission of bioaerosols, as for example, spores. And finally, we also study the soil stabilization of biocrusts and their impact on dust cycling. And with all of that, we want to estimate the relevance of cryptogamic and microbial communities in mitigating global change effects. So all of the, and we want to raise awareness for this relevance, and all of that research is conducted under the umbrella of global change.
Bettina, sorry for 1 second, but you're not sharing your presentation. I don't know if you were meaning to do it so.
Yes.
Okay.
Oh, sorry.
Then please for
I was-
interrupting you, but I wanted to let you know that you were
thank
not
you.
sharing the presentation.
Oh, wait a second. I thought this would work. Let me see. Wait a second. 1 minute. Thanks for telling. Here we are. So you should see it now. Let me see. Okay, now it's
You share
now
the
it's
full
better.
screen. Yes, I think so. Okay,
Okay.
thank you, Bettina. Yeah, you can
Sorry.
continue. Apologies.
So that was okay. Good. Let me see the pointer again. So perhaps I shortly say that once again. So we are interested in cryptogamy communities in general. So both growing on soil, forming biological soil crusts, but also epiphytically on trees and also colonizing rocks. We are interested looking at the methods. You find them on the right-hand side here. And we are also interested in where these organisms occur. So we look at the geographic location, and we are also interested in the habitat conditions under which the organisms occur. Next, we are interested in their relevance. What are they doing and how are they doing that? So we look into biogeochemical cycling processes. And here we look into the water, carbon, and nitrogen cycling. We're interested in the emission of volatile organic compounds and bioaerosol cycling. And finally, we are also interested in the roles of biocrusts in soil stabilization and in their role in the overall global dust cycling. And with all of that, we want to also estimate the relevance of cryptogamic and microbial communities in mitigating global change effects, and we want to raise awareness for that. And all of that is conducted under the umbrella of global change questions. So our research, I want to start out with a very old research study. We started research on biocrusts approximately in the year 2000 in my favorite biocrust study site in Southern Africa in the succulent Karoo. And when we started there, we saw, hey, there are great biocrusts between the shrubs, but there's really this wealth of different organisms, and we didn't really, and many of them you cannot really identify by the naked eye. So we thought, okay, in order to conduct fieldwork, we have to come up with some kind of types of biocrust types. And then we just thought, okay, how about going along succession? So when you disturb a biocrust, you first have bare soil. And after disturbance, then if the conditions are right, then you first have that initial light cyanobacterial crust. And after some good rains, this darkens and you have a dark cyanobacterial crust. And this could further develop into a cyanolichen crust, or also a crust with chlorolichens, with mosses, or with bryophytes. So we really defined these different successional types. But I was not sure, does that really make sense this way? Is this that we just make these groupings because they make it easier for us to work with them, or do they also make sense in a biological way? And then much later, like more than 15 years later, we thought, okay, let's look into these crust types and into their microbial composition. And here we conducted a study looking into the bacterial composition within the biocrusts. And looking at the phylum level, we found that in fact, along this succession from bare soil to chlorolichen and moss crusts, we found that some phyla were increasing in abundance and others were decreasing. So we indeed found some differences at the phylum level. And also looking then at the family level, and here you have again the different crust types on the x-axis, and you see different patterns depending on different crust types. And then conducting an NMDS, you see here that we have a complete change of the bacterial communities from bare soil, here shown in gray, to biocrusts. And we also see alterations from the early stages with cyanobacterial crusts through chlorolichen to moss crusts. So we see that the bacterial community, in fact, reflects this succession. And we also see that here in this image that along succession, on the one hand, the heterotrophic microbial composition changes, but we also learned that also the physicochemical habitat properties like temperature, water, nutrient content, soil texture, they are different for the different crust types. And we then thought, okay, with these differences, there should also be differences in the physiological processes. And we look into that with the next slide. So we knew already, or science already knows for a long time, that biological soil crust fix atmospheric nitrogen. But then we wanted to look into the reactive nitrogen emissions. There are these two compounds, which are nitric oxide and nitrous acid, which influence atmospheric chemistry and climate. And we wanted to know if these compounds, NO and HONO, are also emitted by biocrusts. So we conducted dynamic chamber measurements and did a full wetting and drying cycle. And indeed, we found that different crust types had different emissions patterns, whereas from bare soil, there was only very small amounts of NO and HONO emitted. And we found that also that the soil emission quantities were related to different biocrust types. So we learned from that indeed biocrusts play an essential role in nitrogen cycling in soil, both as related to nitrogen fixation and as related to reactive nitrogen emission. Okay, now we saw, okay, our crust types indeed make sense. But one thing that remained was always that mapping these crusts mapping these crusts in the field was really tedious. We had these grids, and with these grids, we then looked at the coverage, or we did different methods and estimated the coverage of different crust types, and that was really tedious. And there was also some subjective issue that different peoples consider different things. So we thought there must be another way of doing that. And for that, we applied deep learning methods. This means that we first had our data acquisition taking lots of pictures, and then we had to tell our model what the different crust types look like. So we trained this model by means of image annotation first, and then we went through a training process. And then in the end, we could apply this model. So if we look here, we see in the central part, we see the initial pictures, we see the training, the image annotation where we train our model on the different biocrust types on the left-hand side. And on the right-hand side, you then see the model output. So with this new deep learning method, we also used a special method, the joint energy-based semantic segmentation. And this is important because we wanted to have really a robust segmentation method. This means that if you do a monitoring project over long time spans and you have different cameras, for example, you still get reasonable results. And if we look at the results of all that, we see, okay, on the one hand, we can look at species coverage, seeing how many cyano, moss, and lichen-dominated biocrusts we find. But also above that, we can also do a neighborhood analysis, looking what grows next to what. And we see that the preferred neighbor indeed are cyanobacteria. Most organisms, and especially most lichens and mosses, grow next to cyanobacteria, whereas lichens are only rarely the neighbors. And then we also did an edge curvature analysis, and we learned that for the lichens, they were mostly convex, meaning that they were spreading. And for cyanobacteria, we saw that they most of them were concave, so meaning that they were receding in our imagery here. So we see that this method facilitates the rapid identification, the mapping, quantification, but also neighborhood and succession analysis. A short glimpse into future work. We just established an experiment on the effects of climate change in alpine biocrusts. And if you think about high alpine regions, or also regions where the glacier just retreats, the very first vegetation you find are biocrusts. And this is essential for the vascular vegetation to establish then. So we only could have then vegetation. So biocrusts are highly relevant for the settlement of the other vegetation. On the other hand, we know that climate change, especially, and warming, especially hard hits the alpine region. So this is really a big topic there. And for that, we just established this alpine warming experiment. We also have some climate stations there, and we are really excited what comes out there. And you see that this is also not the worst place to work at. So, and with this, I want to finish. These studies are only possible with a great research group. We also have scientific and technical support, and for sure, financial support is also important. Thank you. That's the end of my presentation.
Many thanks, Bettina, for this
You're welcome.
really fascinating presentation. I really liked it. And also thank you for illustrating us about the key roles of biocrusts in ecosystem functioning, their succession, and also very interesting how we can better map them with these deep learning approaches. I found that really, wow, over the top of my head.
I'm glad.
Yes. Then our next speaker is Emilio Rodriguez-Caballero. Emilio is a researcher at the University of Almeria in the southeast of Spain. His research is mainly focused on the role of biocrusts on key processes governing the functioning of drylands, their interactions with other surface components, and the influence of global change on them. And for this, he combines field studies with modeling and remote sensing techniques, which allows him to address these questions at different spatiotemporal scales. Emilio, please, the floor is all yours.
Thank you, Raúl. So I will share my screen. 1 second. I have to find where is the... It's now working?
Yes.
Okay, so today, I want to give an overview of the role of biocrust, but not only thinking on the processes that happens locally, but thinking on how those processes interact across different spatial scales in order to get a better view of the magnitude of the effect on those processes. Just to start, I have to say that as most of people working in biocrust, we have been interested during the last year in analyzing the role of biocrust on different processes, mainly in dryland, where we are one of the most relevant communities. But if I have to choose some of those processes, I will say that we have been mostly focused on water cycles and related erosion processes. To analyze that, we performed several experimental studies in the field, in the lab, and we found that biocrust modify all the different components of the water cycles. And those effects depend on the composition of the biocrust and also on the underlying soil properties and environmental properties. So, for example, we know that some biocrusts can promote infiltration in soils with low permeability, but they have the opposite effect in sandy soils, with an overall effect of increasing humidity, even in the situation where they promote runoff, and with a strongly decrease of related erosion. So, for example, here you can see some result of rainfall simulation here in Almeria, where we found that all the different biocrust types reduce erosion by almost 1 order of magnitude, which is a very big effect. And these results similar results can be found with different techniques, natural monitoring or a different spatial scale, and in all the different places around the world. So this is a very clear effect and very significant effect. And something similar happened also with wind erosion. These are very recent wind simulation for the last week, with different biocrust types compared with reference soils from many different ecosystems. And again, you can see the strong protective effect of the biocrust, this strong local protective effect. And these are some samples, some bare soil samples before and after the simulation, and the lichens in the same condition before and after the simulation. So all those studies prove that biocrust cover and composition modify all those processes. Those and other processes, as Bettina already explained, like, for example, carbon and nitrogen fixation. And this effect depends on the development of the biocrust community. Normally, more developed biocrust communities, like lichen and mosses, have more important effects on those processes. But we also know that those processes can modify or can influence other components of the ecosystem, like, for example, vegetation, because there are a series of spatial interaction between the different components of the ecosystem. And for that, we cannot stay in the plot scale. We have to open our study scale at the ecosystem level, for example. So we have to choose the proper scale for specific processes. So, for example, in the case of hydrology, the best unit for analysis is the catchment. So, and how we can perform those experiments or evaluate those processes at the larger scale. So for that, we need specific maps of the distribution of the different components. We spent a lot of time during the last year trying to develop mapping methods based on satellite and airborne images. With those maps, we have this spatial information that can be combined with the information we got from the field experiment at the plot scale in order to develop physical models that give us information about the spatial distribution of the processes. Again, if we come back to the example of the runoff generation or hydrology, we can see, for example, this is a simulation of one rainfall event in a small catchment here in Almeria. And you can see these are biocrust dominated areas corresponding with those areas here. And we can see by analyzing this spatial component that most of the runoff in biocrust is redistributed to vegetation. So there are other processes that are not shown in the local scale, like those ecohydrological feedback of biocrust providing water to vegetation for the growing of the vegetation in this ecosystem. We have to think in an ecosystem with less than 300 millimeters of rainfall. So this is a very important ecohydrological feedback at the ecosystem level. And those processes cannot be analyzed if we only focus on one scale. So it's not only the summatory of two different components, but also the spatial interaction and all those potential feedbacks. Something similar happens with other processes, like, for example, erosion or even carbon fixation or nutrient redistribution. But we also know that there are other processes that cannot be analyzed, even at the ecosystem scale, because they operate at larger spatial domain. So this is the example of, I would say, the dust emission. So, for example, we know that dust can be transported hundreds of kilometers. So if we want to really understand the role of biocrust on those processes, we cannot focus on the ecosystem because the effect is even larger than the ecosystem scale. And for that, we have to jump to the global scale. But here we have some more complication because actual mapping methods based on satellite images cannot or do not work very well at the global scale. And for that, we decide to use other methodologies, but it's just a combination of methods. We play at this scale with statistical models, a species distribution model, in order to get information or some idea about the global distribution of biocrust. According to this model, we are talking about 12% of the global land surface. Once we have this spatial distribution in a similar way as we did at the ecosystem scale, we can apply some models to get information about those processes working at larger scales. This is one example. So we can put the information of the wind simulation together with the maps in a system model to get an estimate of the biocrust effect on the global dust cycle. And by doing that, we found that biocrust reduced the emission of 700 teragrams of dust per year, which is a very big amount. It's about half of the total dust that is emitted in the earth annually. And this dust is transported very long distance, interacting with other processes, like, for example, incoming solar radiation or cloud formation. So again, this spatial context is very relevant, and we have to evaluate not only the local, but also the ecosystem and global scale, considering the spatial context where all those interactions occur. Another interaction is the interaction of this emitted dust with other ecosystems after deposition. As we know, dust transport nutrients and organisms that modify the condition in the ecosystem after deposition. Another good point of those spatial distribution mapping based on statistics is that we can combine them with, for example, climatic prediction of the IPCC in order to get an idea how things will be in the future. And we know that those models say that things will be not very, very nice in the future, because according to those conditions and the models, we expected a reduction of the biocrust cover in the future with very important impacts on, for example, the process we showed before, the dust emission with an increase in dust emission and modification of all the other interaction we mentioned before. Something similar happening with many other processes. So this is just an example of similar analysis for carbon and nitrogen fixation. So we see spatial pattern of carbon and nitrogen fixation by biocrust, and we found that it will be reduced. Again, we are talking about an important spatial context because, as you see here, the distribution or the effect of biocrust is mostly focused on drylands where nutrients are limited. So this is an important spatial effect on dryland regions. So what we have learned for those kind of studies, though, we have learned that biocrust play a prominent role in several processes, but we also know that those effects are relevant not only at the local scale, but also at the ecosystem, regional, and global scales, at which other feedback and processes comes up that are not shown when we focus on the local scale. Finally, with the exercise of climatic projection, we also know that biocrust cover loss will result in several alterations at the ecosystem level with potential impacts also at the global scale. So just to finish, I have to say that we have to think on mitigation activities in order to prevent all those negative impacts. So that's all, and thank you very much for the opportunity to give this talk here.
Many thanks, Emilio, for this very insightful presentation and also for navigating us through the complexities of biocrust structure and functioning across scales. Really, really, really interesting, that effect on dust, emission and deposition. And well, let's see how it plays out. Well, our next speaker is Claudia Colesie. Claudia is senior lecturer in plant physiological ecology at the University of Edinburgh, where she studies the role and status of biocrusts in polar ecosystems, aiming to reveal traits that permit lichens and mosses to exist and also persist in extreme environments. She also studies the effects of ongoing climate change and human impact on biocrusts. Claudia, please, when you're ready.
Thanks, Raúl, for the introduction. Hi, everyone. I'll just share my screen. I hope you can see that okay?
Yes.
Excellent. Thank you for confirming. Yeah, hi, hello, I'm Claudia. I study biological soil crusts in polar regions, so both in the Arctic and in Antarctica, and this talk specifically is about them in Antarctica. And let me just start this presentation by introducing you to Antarctica as a continent. Wait a second, why is this not? Oh, there we go. Continent of the extremes, that's what I wanted to say. Antarctica, as you might imagine, is extremely cold. Temperatures, even in the austral summer, are often just around zero. Here you can see our camp in the middle of the summer when we do our research. Temperatures in winter go down to around -60 degrees Celsius. Antarctica is also extremely dry as a consequence of the temperature. And some people say that the surfaces, ice-free surfaces of Antarctica resemble the surfaces of Mars more than they do any other surface on the planet. Antarctica is also extremely dark. Over the austral winter, there's 3 months of complete darkness. And as a complete opposite to that, Antarctica is extremely bright during the summer. Any organism living in this environment will have to endure 24 hours of sunlight being baked under really high UV exposure as well. Antarctica is also extremely beautiful. I bring this picture of this wonderful king penguin chick here just to bring that point across. And it's because of that combination of extremes that when the first people explored Antarctica, like here, for example, Robert Scott and his party, when they explored the South Pole, they found and noted down in their diaries that there lives nothing. Not even a moss or a lichen is what they specifically said. And I'm happy to say that we do know better now. as part of my PhD and many other people's work, we do know now that Antarctica, in fact, is the continent of biological soil crusts. It is dominated by this community. It is, and they are composed of a diverse area of lichens, mosses, liverworts, and they are dominating the vegetation and really kickstarting every ecosystem process in Antarctica. There are also 2 native species of vascular plants, but they play a minor role when it comes to ecosystem functioning down south. What I study in this context is the physiology of those biological soil crusts, individual organisms, but also the crusts as a component. So I try to understand how they function, how they work, how they live and survive this combination of extreme conditions. I study the taxonomy of the biocrust, trying to understand which species are where and how they are distributed. And then bringing this together, I try to understand how they make the ecosystems function and what they contribute to wider carbon and nutrient cycling. And in terms of this ecosystem function, it's important to mention that, of course, the polar regions are currently undergoing significant change, especially in the polar regions. We have very severe effects of global climate change already happening. And this is, of course, also affecting the vegetation and the biocrust in Antarctica. And this slide, without going too much into the detail, really should just show that there's a multitude of threats currently happening all across the Antarctic continent. And those threats are very diverse, and they are also regionally different. So depending on where you are on this big continent, the effects of climate change will be different, and the responses of the vegetation to those changes will be different. And it's striking to think about that in the context that here we talk about a system where we still don't know exactly where things are. So here I want to show you this project where this was an expedition I was on exactly 1 year ago. I was sailing exactly the waters you can see here around the South Sandwich Islands. This is an archipelago of 11 volcanic islands in the middle of the Southern Ocean, and it is a complete white spot in terms of our knowledge of terrestrial biodiversity. We do not know which organisms live on those islands, neither plants nor invertebrates. And it was really challenging, really interesting trip. I've produced a little video about it. If you scan the QR code up there, then you can have a look at how we managed to navigate these islands. And what I want to say is that it's really difficult still in this context to know where things are, and these expeditions are expensive. These expeditions are very difficult to manage and logistically plan. So one way we could look at distribution of vegetation in Antarctica would be to use remote sensing. And here I want to introduce you to this work, which is led by my current PhD student, Charlotte Walshaw. She is using remote sensing technology. This is Sentinel-2 imagery to try to detect where organisms grow in Antarctica. And this is the first map that we have, in fact, on a continental scale that outlines where we find green vegetation, so combination of vascular plants and mosses, where we find lichens, and where we find green snow algae. And the major take-home message from this is that there is about 44.2 square kilometers of vegetation in Antarctica. 44.2 square kilometers, that is about the size of Manhattan on a continent that is larger than the former USSR. So it really is not very much. It is a tiny proportion of Antarctica that is vegetated. And that underlines how important it is to conserve it and to protect it in any future scenarios. And this map is so important because it provides us with a baseline. Now we know where things are, and we can then now start monitoring how things might change in the future. And in fact, all across Antarctica, we only have 3 sites where we have long-term information about vegetation dynamics in the past. This is one study that I just want to show. It was initiated by this professor called Emanuel Rudolph, Ohio State University in the US. In 1961, he went to a site called Cape Hellett, and he drew a very detailed map of where he found lichens and mosses and biocrusts in a plot that is 30 meters wide and 120 meters long. And I was lucky enough to go back to this particular site almost 60 years later and survey and find in the soil, still buried, the original pegs that Rudolph used to outline the shape of his plot. And that allowed me to completely resurvey where the lichens and mosses have, where they are right now. And I did this based on a ground survey, but also based on a drone imagery. And that allowed us then to create a much more detailed map of where we find mosses and where we find algae, cyanobacteria, and lichens in this plot. And by analyzing Rudolph's original map and our new map, we then found that in fact there is not much change. These mosses, although we have records over 60 years, have not grown much. And that just shows how incredibly, incredibly slow these organisms work and what their natural functionality is and what that means in terms of their fragility, in terms of fast change happening in the future. If you're interested in this kind of research, my research group, we call ourselves the cryptogamiacs because we are manic about cryptograms. We have a web page if you want to look us up. All of this research would never have happened without very, very many wonderful people, amazing support from different national polar programs and very many different people across the planet. Thank them all very much and you for your attention. Thank you.
Many thanks, Claudia, for the very, very nice presentation and also for allowing us to marvel at the uniqueness of biocrusts from Antarctica. Very beautiful, also very extreme, right?
Yes.
Then, totally, yeah. Our last speaker is Matthew Bowker. Matthew is a professor in the School of Forestry at Northern Arizona University, where he also serves as Associate Director of the Center for Ecosystem Science and Society. He defines himself as an ecosystem ecologist drawn to the soil. He's particularly interested in understanding the dry corners of the world where he has lived most of his life. And despite this lifelong relationship, he has never stopped being fascinated by the mini ecosystems that are biocrusts. This has also sparked his interest in developing strategies that allow their recovery when they become degraded. Matt, please, the floor is all yours.
Thanks, Raúl, and thanks everyone for listening, and thanks to GSBI for this great opportunity to talk to you. I am, as Raúl said, I'm interested in just about everything about biocrusts, but one of our focal areas for the last several years in my group has been finding ways to restore biocrusts after they've been lost from degraded ecosystems. You've heard a little bit so far about the many important functions that biocrusts perform in ecosystems. Things like soil stabilization and nutrient cycling and hydrological effects and vascular plant interactions. And you may wonder, what can't they do? They seem to be good at everything, but they are bad at one thing in particular, and that is resisting disturbance. And when I'm talking about disturbance, I mean any physical surface disturbance, like you might find from the impacts of livestock grazing, or these surface disturbances related to energy production, or a natural process like fire. And when you have a disturbance, you can lose the biocrust very fast in minutes or days. But then the recovery part often takes years to decades, and maybe longer in some extreme environments like Antarctica. So my group is called the Dirt Lab, and the research that we do is aimed at bringing back the biocrust, finding the way to do it. And our basic game plan is to grow the biocrust organisms in artificial systems. And then we take that biocrust that we grew and we add it back where it's been lost. And then we try to encourage the recovery and establishment of the biota, of the biocrust biota. And in the process, our aim is to heal degraded ecosystems. First, growing biocrusts. It may be surprising that you can grow them. We've just heard how static they are in Antarctica. Over 60 years, nothing happened. But I want to stress that slow growth is not hardwired into the organisms. Slow growth is caused by the environmental constraints. And if you relieve those constraints, you can grow the organisms remarkably fast. One approach that some research labs around the world take is to go into the biocrust, find particular strains of cyanobacteria, for example, and then try to culture them in liquid media, which is basically a nutrient soup. And this kind of process for some species has been done at a fairly large scale, like pretty big ponds. Another approach is instead of isolating community members, try to grow the whole community. This is something my group's been working on a lot lately. We create these outdoor farms with irrigated beds, and we have to add a little bit of starter material of biocrusts, and then we irrigate it and turn it into more biocrust, much more. Another approach might occur in the greenhouse. So we've developed a few greenhouse methods for growing biocrusts on soil, the whole community. One method invented by Kyle Doherty exploits the ability to harvest fog by certain biocrust organisms like these mosses. And just with fog alone, we can irrigate and grow biocrusts. And in a couple of months, basically you've grown little pieces of biocrust moss lawn that you can then harvest and apply to degraded field sites. So the first overall finding that I want to share is that you can grow biocrusts, and generally speaking, cyanobacteria and mosses can be grown. The lichens are definitely more difficult. There's some exceptions and systems where you can grow some lichens, but the lichens are much more difficult. So once again, we can grow biocrusts, and there's different ways that different labs use to do it, but that's not the end goal. The end goal is to then take the biocrust biota that you grew and then establish them in the field in degraded sites. And that's easy to say and hard to do. And some of the challenges that you encounter are harsh environments, or maybe the biocrusts that you grew lost their field hardiness. And there's a lot of loss mechanisms. Wind and water can just sweep away the biocrust that you had before it gets established. And we've even seen insects carry biocrust away before it establishes. So the second key finding I want to share is our current focus. What we're finding lately is that integrating three things can be really helpful to actually establish biocrusts in a degraded field site. The three things are the addition of the biocrust biota at the same time as softening the environment, at the same time as decreasing the losses of what you're adding. And if you can do all those three things in the same moment, this will lead to better establishment success. And we've been trying to integrate this in this kind of the same technique or the same physical object. And some examples are techniques we call biocrust cloths, sods, and pellets. So I'll show you a little bit about that. So this is Kyle Doherty's moss cloth. He used that fog chamber I showed you earlier and actually grew a biocrust moss directly on the cloth, no soil whatsoever. And then that cloth becomes this physical object of the moss and the cloth, which is sort of a protective shield that buffers the environment. And you can attach that to the soil with a mossy side underneath. And this can lead to establishment of a biocrust patch. So that's one way we're working on this. A somewhat similar but distinct way is what we call biocrust sods. And we've got a few people, Kevin Griffin and Lydia Bailey working on this, and Colin Tucker did a lot of early work on this. And basically we're using these outdoor farms and we're creating these beds of soil underlain by different kinds of cloth. And we grow out the biocrust for a few months, let it attain high abundance and high development. And then here's a bunch of people harvesting some biocrust sods. They're cutting it into 3-meter strips, wetting it up. And then when in the wet state, you can actually roll it up without tremendous damage. And that can be transported to a field site. And then at the field site, it's just like unrolling a carpet. You just you carry that out where you want to put it, unfurl it, and instant biocrust. And you can see those green dark patches that this is a well-developed crust. This kind of product not only establishes biocrust, but if we put a weed barrier, a biodegradable weed barrier on the bottom, it can also be a way to fight unwanted flammable weeds, for example. And then the last one, my student Jasmine is working on a lot lately. And the idea here is to impregnate these protective pellets made out of a material like clay and put biocrust organisms inside. And then as the pellet degrades, it's a slow release of biocrust organisms into the environment. And we found that this can lead over between 1 and 2 years to pretty strong biocrust colonization for certain types of biocrust. And here's just a close-up of a really successful example of that. So what's left? We don't know a lot about the long-term outcomes of these methods. We only know sort of usually a couple of years, maybe up to 4 years. And we don't know particularly how well these methods can scale up to hectares or even square kilometers. Maybe a bigger question is, should we be thinking about those kind of skills at all? And maybe we don't have to. I'm glad to talk about that if you want to. One last thing, another interest of mine is globalizing biocrust ecology. And I just wanted to plug a project coming up over the next few years. It's called CrustNet. And if you want to understand what it is, just imagine what if all the biocrust researchers did the same set of ecological studies together and then put all the data together. And we're about to find out what happens. So thank you for this opportunity.
Many thanks, Matt, for such a wonderful and insightful presentation. I very much enjoyed it, and I'm sure that the audience did it too. Actually, I was particularly intrigued by CrustNet, of which I'm sure we will hear a lot more in the not so distant future. Now we will move to Q&A. And if people don't have many questions, then we'll come back for sure to the CrustNet. So you will not escape. Okay, let me just check the Q&A box. For now, I see that we don't have many questions. So, Matt, perhaps this is actually a good moment, now that you have mentioned it, to start talking about CrustNet. Please, everyone, all the panelists, join for the Q&A and for this part of the webinar, where we will talk about different things. We will answer questions. We will share, well, our experiences with biocrusts. Well, perhaps, Matt, before you start, I would like to thank all of you again for these really wonderful presentations. I think that they were very insightful and also highly complementary. And I think that's really good, because now we can have a very wide view of the research that is currently being done at the edge of the research on biocrusts. Okay, so please, Matt, if you don't mind to answer my question.
Sure. So CrustNet is an example of what's called a distributed networked study. And the idea here is that we develop a set of studies and protocols and methods, and then ask people around the world to repeat the same study using the same methods. And then together we build a global data set. And what CrustNet is built to do is it's kind of meant to be a multi-purpose everything study about biocrust ecology. However, there are a few focal areas related to functional diversity. In particular, we want to learn what are the global controls on functional biodiversity in biocrusts. And we also want to learn what are some of the outcomes of functional biodiversity. For example, does it increase ecosystem stability? And this will be launched in 2025. Our funding will kick in January 1st, New Year's Day, and you'll be hearing from us about it. The principal investigator team is myself, Anita Antoninka, Javier Sejan Navarro, Sasha Reed, and Anthony Derazet Nardi. And we've already attracted excellent postdocs here at JETCH that will help us get rolling.
Thank you, Matt. Actually, we have our first questions coming, and one of them was related to CrustNet by Diane Hockland. She was asking if there is still time to get involved in CrustNet. You said that it's starting in 2025, so I guess that there's still time to get involved. And yes. I don't know if you can tell us a bit more on how to get involved, or maybe that's something that we have to wait for.
Yeah, right now, wait, and we're going to distribute the word widely through our website. We've already built an early version of the website, and we'll be posting protocols, and we'll be doing email blasts on Blue Sky and Twitter to make people aware. You'll hear about it. We'll make sure you hear about it.
Okay, awesome. Thanks, Matt. Well, we're getting a lot of questions, but specifically all for you, Matt, regarding your approaches also to recover degraded ecosystems. Gonna pick one. Sankar Manoharan is asking if artificially grown biocrusts have similar communities to the original community. So are they equivalent, the ones that you're growing in the greenhouse as compared to the ones that you find in the field?
That's a great question. So when the researchers that grow one organism at a time, what they do is they separately grow the organisms, keep them in pure culture, and then try to create combinations at the end to add back that resemble the original community. But when you're growing the whole community at the same time, like my group often tries to do, you do get some drift. You get some members of the community that don't grow so well, like most of the lichens usually just sort of hang on or maybe slowly die, whereas the cyanobacteria and the mosses reach elevated levels. So it does distort the proportions of the community a little bit. And that tends to then be filtered by the environment when you put them back outside.
Okay, thank you, Matt. I have another question now for Bettina. This is a question from Cecilia Floco, who asks, how does microbes are equated in the machine learning process based on images? So in other words, are you considering them in your deep learning approaches?
Yes, that's a very good question. Unfortunately, we cannot identify the microbes in these images. So we see really the photosynthetically active compounds. We see the lichens, the bryophytes, etc., cyanobacteria, but we cannot tell from the image which microorganisms are there. So for that, we would then need to link the imagery then with microbial analysis in order to learn more about that. But very good question. Thank you.
Thank you, Bettina. And I have another question that perhaps may be answered by any of you or by all of you, depending on your different experiences, because maybe this is something that varies by ecosystem type. So the question is by Arturo Jimenez Martinez, who wants to know how deep can a biocrust become in natural ecosystems? So perhaps any of you can answer. Maybe Claudia, starting in Antarctica.
Yeah, it's an interesting topic because of the way these organisms grow. So a moss bank, if you consider that a biocrust, can grow really, really thick, maybe a couple of meters even. In the Antarctic, we have some mosses that are 30, 40 centimeters long, an individual shoot. That is still a biocrust per definition, as it's a pure moss carpet. It's a good question to ask, but definitely they can accumulate a lot of biomass and they can store carbon for a relatively long time if left undisturbed.
I think I can also add something to it. So there have been quite some discussion about that. What is still a biocrust and what is not a biocrust? And we discussed a lot about that when we met, when we had together that one paper with many of us, and also Matt was really important in that. And we discussed a lot and said, okay, it's really that the main part of a biocrust normally is below ground and the smaller part is above. So yeah, but it's kind of fluent. I would also say the thickest crusts, in my opinion, I see the pinnacled crust. This is because they have this freezing and thawing events. And so you have really these pinnacles, you have that they grow really 3D, and these, in my opinion, are really thick crusts. But then they still are like up to 10 centimeters or so, I would say. This would be my opinion, but the others, please jump in and add your opinions on that.
Yeah,
Emilio, sorry.
I'll jump in a little bit. I think we did write this paper a couple of years ago, and it's called "What is a Biocrust?" if you want to look it up. So that's sort of a synthesis opinion of a bunch of us. We didn't really want to draw hard boundaries between a biocrust and not a biocrust. We just wanted to provide a guide to when people are talking about biocrust, generally, what are they talking about? And most of the literature examples are quite thin. They're usually less than a couple of centimeters, maybe only a millimeter or two thick. But we fully leave open the possibility of a thicker example like Claudia or Bettina were talking about.
Emilio, I don't know if you want to add something to it.
I think
based on your experience in south of Spain.
I think everything is explained now, so but I agree with the definition, probably providing this study of biocrust definition study of a few centimeters in the biggest class. But of course, we have to take also care about the biggest mosses. But probably we have to add that it's not only the depth of the biocrust, but also the influence of the biocrust below the class. So probably we have to extend the ecosystem to some centimeters below the class where the effect of the biocrust is still visible at different levels.
Yeah, I think that's a very important point. Actually, related to the same to the question, there's another question by Arturo who asks if someone has done studies through the soil profile with thin sections. And I think that's really related to what you're saying, Emilio. It's not just the biocrust, which may be just a thin layer, but what's underneath, and that may be more related to the composition of the microbial communities, right? I don't know if you can say something about it.
So here in our group, I do not do this thin profile, but some of our colleagues did several years ago. It's again the same. So we have the 1 to 2 centimeters of the effect of the biocrust in the more developed one, and some other effects in the porosity, so centimeters below. And I think there are some people in the public that makes 3D analysis of the porosity of the class, but not in the panelists.
Mm-hmm. I can also add something. So we did some cross sections of biocrusts and then looked into really the composition. We used also fluorescence in situ hybridization fish to look at the different organism groups. And what we saw was that really biocrusts, if you look at them in this cross-section, that the different organism groups are distributed very heterogeneously, and also the pore spaces and the texture is really heterogeneous. And for us, this was really important because when we looked at our nitrogen gas emissions, we found such different patterns depending on the exact spot where we were measuring. And then looking at these cross sections, we understood, okay, there's really different things going on depending on the exact spot where you are measuring. Yes.
If I could just add to Arturo's question, there are indeed a couple of people looking into the effects of the biocrust organisms below ground and doing all sorts of cross sections, but also longitudinal sections. And the most interesting organisms, I think, in that context are the cyanobacteria, because they have a sheet of sugars around them, and this sheet of sugars, they get thicker when they're wet, and then they shrink when they dry out, and that has effects on the stability of the soil. So the researchers looking into that mostly are interested in soil stability and erosion effects and in penetrability of the soil.
Thanks, Claudia, for your answer also, for also adding that. Then I think we're getting to the point that we're understanding that these biocrusts, they're not only a mixture of lichens and mosses plus cyanobacteria, they also contain a lot of microbes, including fungi, bacteria, but they also contain other organisms such as protists, right, that are part of food webs. They are complete food webs with herbivores and predators and decomposers. And so in relation to that, Natalia Zibinska is asking if there's any research going on about how biocrusts interact with soil invertebrates like Oribatida and other mites, and also colembolans. Can some of you reply to it?
I can start that one, because in Antarctica, where we don't have many animals, and it's in fact the colembola and the mites that are the largest animals that we find in many of those places, we have relatively disconnected little food webs that we can study really well in terms of what is the effect of the biocrust on the existence of these animals. And it's really striking to see that some of these tiny little mosses are full of colembola, and they are hiding inside, and they are feeding from the cyanobacteria that grow on the top of the little moss leaflets. And if you just step 1 meter away, it's completely sterile and barren soil. So the biocrusts in the Antarctic really kickstart this ecosystem functioning by providing food sources for the tiny little invertebrates.
I perhaps can add a little bit to that. So in fact, studies on invertebrates in these food webs, I think they are really rare, and this is still a field where a lot more research could be done. There are some names who are active in that, and the names popping into my mind are, for example, Brian Darby and Deborah Neher, or on the European side, Kenneth Dumac. He has been looking at protists. But in fact, this is really a field. We know there's a lot going on. And I think this field really deserves a lot more research, and would be interesting to have more people attracted to that topic, I think.
Bettina said exactly what I was going to say.
Okay, thank you. So there's another question by Tiago Enriquez, and I think that this could also be answered by anyone, perhaps by Emilio, because it's more addressed to the global scale. So the question is if there are regions in the world where studies on biocrusts are particularly scarce, and also adding to that, how you think we can change this paradigm.
I think, yes, there are many places where we don't have information. This is something that we learn when we make this global map, comparing all the different studies. We found that some regions, like for example, South America, is not well understood. And also in Africa, we have many places where there is a lack of knowledge. So probably relates with the lack of resources for research. So most of the research is focused in Australia, China, probably in the US. Maybe CRUSTNet will help to solve this limitation in information in many regions.
That's a very good point, Matt. So what's your goal regarding the spatial coverage of the CRUSTNet?
Yeah, it should help fill in some gaps, and we'll, so far, if you want to understand spatial coverage, you have to rely on literature data that's collected with different methods and the idiosyncrasies of different groups and the different things that they do. But CRUSTNet would provide at least a decent number of sites that are all measured in the same way. So I think that could be really helpful.
Thank you, Matt. I have another specific question for Claudia. And the question is from Cecilia Floco again. And it is, what is the resolution of the remote sensing method used for the vegetation survey in Antarctica? And if there are confounding factors that have to be filtered out? Claudia?
Thanks for that question. If that refers to the continental scale map, the pixel size is 10 by 10 meters. So it is really tricky to detect these tiny little organisms on a pixel that is that big. So in fact, there were a lot of constraints to overcome, and the methodology worked best when the pixel was covered by green vegetation. So when it's with mosses and vascular plants, because that's more easily detected. If it was mixed the vegetation then with lichens, then it was harder. So we had to use different indices to detect them. And then there was a certain threshold when we said, okay, now this is too uncertain. We'd rather not count it as vegetation than we count it as vegetation. So it's probably an underestimate, our estimate that we provide with that map.
Thank you, Claudia. We have a question from Yuri Dimitruk. And the question is if there are aspects of using biocrusts for remediation that could be used to restore sites that have been degraded by bombs, mines, missiles. I guess this can be used also, or it can be extended in general to sites that have been heavily degraded. Matt?
Yeah, I don't think anybody has ever studied that specific test case, but my answer is yes, I think so. Any kind of disturbance, there's a wide variety of settings where biocrust rehabilitation or restoration might be useful. Post-fire, after the kind of disturbances you're talking about, for example, on military training lands or areas that are scarred by past conflict, mining of mineral resources, that's another setting, erosion control, anything like that. And, there may be some very creative things we can do with them, for example, protecting archaeological sites.
Maybe if I could add a little anecdote to that, because I'm originally from Germany, and it's not necessarily an environment where we have biocrusts in a natural setting because of the moisture levels. But we do find biocrusts in former military sites. So where the soil has been massively disturbed by tanks driving over the vegetation, that's where we find biocrusts now. So that's kind of linking a little bit to that question, saying that, yes, it seems possible that they can be the first successional stages in such heavily disturbed sites.
That's a very interesting point, Claudia. We have a question from Carmen Rosa Carmona, and she's asking what are the effects of climate change on biocrusts from tropical regions. Actually, I think that we have not really talked too much about biocrusts from the tropics. We've been focused on biocrusts from mostly arid ecosystems or extreme environments like Antarctica. So perhaps before we jump into the effects of climate change on biocrusts from the tropics, perhaps someone could say something about just this type of biocrusts and where we can find them.
Maybe Bettina is the best. It's the closest one to the tropics.
Not at the moment,
Go
but
for it.
yeah, not at the moment, but with research conducted in Brazil in the tropical rainforest, perhaps I can say something. So as long as you have tropical rainforest, for example, you only find biocrusts at spots where the rainforest is disturbed. So for example, when we drive to our research site, where we first land in Manaus, and then we have like a 4-hour drive, and on the roadside, on the side of the road where there is no dense forest, there we indeed find really very nice biocrusts. And yeah, but if the forest is too dense, then there is just no place for the biocrust to occur. But also in other places where the forest has been cut, there people find, for example, in sandy lands, in sand dunes, that they are really then colonized by biocrusts. And a student of mine, Carla Weber, she has found really nice biocrusts, which also stabilize the soil there. So we know they occur there if there is the place for the biocrusts to grow. But I would say we have no knowledge on the effects of climate change. In fact, really knowledge on climate change effects, is based on some experiments, and very recently it's also based on field long-term field studies where we see, okay, biocrusts coverage is decreasing, or we also see a reverse succession, that biocrusts go back to early successional stages. But I don't know of studies for tropical regions where we know exactly how biocrusts do under global change. Perhaps someone else has more to say there.
I can't say much about the tropical case specifically. I'm not sure there is a global change study in tropical, or at least wet tropic regions, maybe in the dry tropics. But I can say a little bit about global change factors and biocrusts in general around the world. There have been a handful of experiments that are imposing treatments like rainfall reduction, or different methods of warming, or different modes of precipitation simulated. And in those studies, the biocrusts do seem to be responsive to the overall rainfall received. And these are dryland systems, so the answer in tropics could be different. But in the dryland systems, the less overall rainfall does lead to a reduction in biocrusts, and warming is actually faster. It's a faster negative response than droughting. And then the fastest of all that I know of in the literature is when the precipitation is changed so that all of the rainfall pulses are very small, and you only get short hydration pulses. That can be positively lethal for certain biocrust members like bosses. A lot to learn, and we need more studies in more areas like the tropics.
So thanks, Matt, also for expanding on the effects of global change on biocrusts. There's a question. This is an anonymous question, but this is actually aligned also with a question that I have myself. And the question is, why should biocrusts not be induced and spread? And for this, I also have a reflection to share with you, and it's that we know that glaciers are fundamental for the functioning of ecosystems and for soil organic matter formation. And we also know that biocrusts, they're very sensitive to trampling by glaciers. So the question would be how to reconcile these views. And also, perhaps a at a larger, in this case, temporal scales, is that the biocrusts, they have survived the trampling of the dinosaurs, the megafauna, etc. So perhaps they're not so much at risk right now. I don't know. What do you have to say about this?
So I can say, for example, that so it's true that biocrusts coexist with many animals in the natural condition. But normally when people describe some important negative effects on the biocrust is when you have an intensification of the animals. So probably when you have some kind of overgrazing or when you increase the number of animals. And this response is even not linear. So there are some thresholds above which the effect of the animals is even bigger. So it's true that they can coexist with the animals. They naturally do during the history. But the problem is that we are increasing the pressure. And this is the problem. So when we cross some threshold of the grazing pressure or animal pressure on the ecosystem. This is, well, to my point of view, of course.
Absolutely. I think that makes a lot of sense. Matt, I don't know if you want to add something to that.
I thought that was a great answer. In most of our livestock production systems, the biomass of the animals is greater, the intensity of the grazing is greater. It's just much larger than what would be occurring with native grazers. So it's not so much that biocrusts are 100% incompatible with herbivores, large mammalian herbivores. It's that beyond a certain density, they can be erased, and they have been nearly erased from large portions of our rangelands.
Awesome. Thank you for your answers. I think we're approaching the end of the webinar, so I have a last question for all of you. And you all have an extraordinary trajectory studying different aspects of biocrusts and biocrust ecology. So I would like to know, what do you think is the greatest challenge ahead in our quest for better understanding and making practical use of biocrusts? Perhaps Bettina can start answering.
Yes, so I think a really big challenge, or one of the major challenges, is really looking at global change, which is both climate change and also land use change, land use intensification. And we know that land use intensification will kick in even more. So we will have a higher demand on land. And then we have this problem: how can we still keep the biocrusts and keep their ecosystem services? And also under the climate change effects. I think this is really a topic which is highly demanding and highly important with all the ecosystem services provided by biocrusts.
Awesome, thanks. Perhaps, Emilio, now you can answer to my question.
So after the answer of Bettina, so I will say that maybe one of another important is that we have to work in the future is in a way to implement or to continuing with the topic of my talk, to try to upscale all the different processes and functions that we know that happen at the very small scale, from the microscopic scale to the ecosystem, to try to put them into this Earth system model, to include in the global modeling in order to get a global picture of the role. And with this, we can get some arguments to try to convince people about the importance of biocrust, which answer the second part, which is the how to make a good use of the biocrust. So we need to convince people to make this good use.
Awesome, thanks, Emilio. Claudia?
Thanks. It's a really good question, and I'm glad you gave it to us before so we could think about it a little bit. I think the greatest challenge is opening up the biocrust research to the next generation of scientists, of students, and for more representative global distribution. I think we are still a niche topic. We're easily overseen and overshadowed by large-scale global scientific endeavors. I think I see this in my daily job as a lecturer in the university. Programs and courses get streamlined more and more. Everything's trying to be much more efficient, but to become a biocrust researcher, you have to you need time, you need passion, and the information we get is not easily streamlined. We have to get bits and pieces from here and there. It's still kind of a developing scientific study. And I think we are a wonderful community. We are genuinely supportive of each other. And I just hope that more researchers explore this area. And I hope that this webinar will help to encourage a few people to come and try to study biocrusts.
Absolutely. That was a very motivating and hopeful response. Thank you, Claudia. Matt?
You're
Matt,
muted,
you're
Matt.
muted. You're muted.
Sorry. I agree with everything that everyone just said. That is a great short list of priorities, better understanding global change effects on biocrusts, really getting to the Earth system scale on biocrust processes, and expanding our research community and overcoming the fact that we're still a niche topic. But maybe I'll add something about the restoration. I think the greatest challenge really is learning how to translate restoration research into actually engineering ecosystems at meaningful scales. And that's a tough challenge, because the scope of degradation is large, and a lot of the methods we're using are, little tiny test plots, and bridging the gap one way or another is a tough thing to think about. But I think there's a lot of roles for creativity, and lots more researchers working on that problem. And I also actually think, this might sound like science fiction, but I also think there could be a role off our world for biocrusts. For example, conditioning Martian regolith to grow food for humans, for example. So I think there's a interesting future in store for the line of research that we're trying to put together for biocrust restoration.
Matt, I have to say that I'm a huge fan of Isaac Asimov, and I completely agree with you regarding the colonization of Mars regolith. That was a very interesting answer. Thank you for saying that. Well, after this really intense and fascinating 1 hour and a half or so, in which we have learned a lot from our panelists, I would like to take the opportunity to thank everyone for your attention and for your questions. And I will now hand the control back to Bim and Lina in the headquarters of the GSBI.
Yeah, thank you very much, Raúl, for chairing this exciting meeting. Actually, it's really fascinating. And Bettina, Emilio, Claudia, and Matteo for all the great presentations. And I saw a lot of nice concepts coming along, from succession to restoration, global dust. It was impressive how much global dust it is preventing, actually. And also, I see opportunities for above-below ground interaction work to do on biocrust, because there's a lot going on subsoil, what I've learned. And even we can go out of outer space, doing outer space research. Although I would make a plea for preventing the biocrust degradation. So let's cherish what we have here on Earth before we go out in space. Yes, this was a great one. I hope you will be also represented at the Global Soil Biodiversity Conference between 12 and 15 April in 2026. Actually, you had a great audience, and I have no doubts that many people will look back to the recorded version because the word will spread. I thank Lina a lot for putting this all together. She's always the science force. So Lina, maybe you can just put up the camera, or yes, okay, there you are. Big applause for you. Thank you very much. And Well, we are just approaching the end of the year, so I wish everybody a great, if you have that end of the year holiday, and a great start for the new year. And I'm looking forward to new webinars that we will organize by then. Thanks the audience for all the great questions. This was super. Thank you very much, and wish you a very nice, well, day, afternoon, evening, night. Bye-bye.
Bye.
Bye-bye.
Thank you. Bye.